We live in a remarkable time. We are beginning to understand the very building blocks of life itself, our genes. And with that knowledge comes incredible power, the power to heal. Gene therapy offers a way to treat diseases at their root by fixing faulty genes. But how do we deliver these therapeutic genes to the right cells in the body? This is where adeno-associated viruses, or AAVs, come in. AAVs are like tiny delivery trucks for genes. They are viruses, yes, but harmless ones. Scientists have stripped them of their ability to cause disease. Instead, these viral vectors are engineered to carry therapeutic genes. Think of it like taking the engine out of a car and using the chassis to deliver packages. Over time, scientists have made these AAV delivery trucks even better. The first generation of AAVs was like the Model T Ford, reliable but basic. Then came the second generation, sleeker and more efficient. Now we are entering the era of third-generation AAVs, engineered for even greater precision and effectiveness. Let's take a closer look at how these remarkable gene delivery vehicles are built. Imagine taking a virus and carefully removing its harmful parts, leaving only the shell. This shell, or capsid, is what we use to create recombinant AAV or RAAV. Inside the RAAV capsid, we package the therapeutic gene we want to deliver. This gene is like the package being delivered by our delivery truck, but it needs an instruction manual, a promoter, to tell the cell how to use the gene. And we can't forget the inverted terminal repeats, or ITRs. The ITRs are like the address labels on the package. They tell the cell's machinery where the gene starts and ends, ensuring it gets delivered and read correctly. Without the ITRs, the gene would be lost in the vastness of the cell. In the world of gene editing, CRISPR-Cas9 is a revolutionary tool. Imagine it as a pair of molecular scissors that can precisely cut DNA. This technology has been instrumental in improving the efficiency of AAVs for gene therapy. CRISPR-Cas9 allows scientists to engineer the AAV capsid, the outer shell of the virus. By making small changes to the capsid's proteins, we can direct the AAV to specific cells in the body. This is like putting a GPS system on our delivery truck, ensuring it reaches the right destination. This targeted approach is crucial for maximizing the therapeutic benefit and minimizing potential side effects. For instance, if we want to treat a genetic disease affecting the liver, we can engineer AAVs that specifically target liver cells. Section 4. A virus's journey entering the cell. Once the engineered AAV reaches its target cell, it must overcome another hurdle, entering the cell. This process begins with the AAV binding to specific receptors on the cell surface. Think of it like a key fitting into a lock. One of the key receptors for AAV entry is heparan sulfate proteoglycan, or HSPG. It acts like a docking station, anchoring the virus to the cell surface. Once attached, the AAV is engulfed by the cell membrane, forming a vesicle. This vesicle then travels to a compartment within the cell called the endosome. Inside the endosome, the AAV undergoes a series of transformations that allow it to escape and finally deliver its genetic cargo to the nucleus, the cell's control center. Section 5. Outsmarting the immune system. Challenges and solutions. Our immune system is a powerful defense mechanism. It protects us from harmful invaders like bacteria and viruses. However, this protective instinct can pose a challenge for gene therapy using AAVs. The immune system can recognize AAVs as foreign and mount an attack, neutralizing them before they deliver their therapeutic cargo. This immune response can limit the effectiveness of gene therapy. To overcome this challenge, scientists are developing innovative strategies. One approach is to use low-prevalence serotypes of AAV, which are less likely to be recognized by the immune system. Another is to cloak the AAV with a protective layer of polyethylene glycol, or PEG. This pegylation can help the AAV evade immune detection.